The National Microbiome Data Collaborative, or NMDC, has developed bioinformatics workflows to process multi-omics microbiome data in a standardized and reproducible way. The NMDC has developed metagenomic, metatranscriptomic, metaproteomic, metabolomic, lipidomic, and natural organic matter workflows that incorporate state-of-the-art tools and algorithms. Each NMDC workflow has been carefully curated and optimized, as well as extensively tested across diverse microbiome samples. The workflows are identical or closely based on production quality workflows currently in use at the Department of Energy user facilities, the Joint Genome Institute, and the Environmental Molecular Sciences Laboratory. Many of these workflows are available through the NMDC GitHub or Docker Hub, which are linked here. NMDC EDGE is an open-source, user-friendly web interface designed to make the NMDC bioinformatics workflows accessible to any researcher that is interested in processing their microbiome data, no matter their experience level. This video will walk you through the basics of NMDC EDGE and how to process data through the NMDC workflows. If you'd like to learn more about NMDC EDGE or our workflows, visit our documentation site here or check out our publication. Here is the URL for NMDC EDGE. And typing that in leads to this homepage for NMDC EDGE, where you can then select in the top right corner to log in with your ORCID information. Then from the navigation bar on the left, you can check out the tutorial section where you will find videos like this one on how to run the workflows and interpret the results, as well as PDF versions of the training materials, translations of the user guides into multiple languages, and additional documentation for each workflow. Moving down to public projects, here you are able to see any NMDC EDGE projects that have been shared publicly. Next, there's the Upload Files button. Users can drag and drop or browse for files to upload. File restrictions in terms of size and file type are described here. Files must first be uploaded here before they can be run through any of the NMDC workflows. Data that has already been uploaded to NCBI's Sequence Read Archive, or SRA, can also be directly accessed by navigating to the Retrieve SRA data page and entering an accession number. And finally, there are the workflow sections. Currently, the metagenomic, metatranscriptomic, metaproteomic, and natural organic matter NMDC workflows, as well as the GenoMAD workflow for identifying virus and plasmid signatures, are incorporated into NMDC EDGE. Information on how to run each workflow can be found in the workflow-specific user guides. As an example, to run the metagenomics workflow, a user can select metagenomics, then run multiple workflows, which consists of running the reads QC, read-based taxonomy classification, metagenome assembly, metagenome annotation, viruses and plasmids workflow, and metagenome assembled genomes, or MAG generation workflows. To run a workflow, a project or run name is required. Then a project description is optional but recommended. Then you can select any relevant fields and select either data that has been uploaded or a publicly available data file, such as those included in the test data folder. Then you can click Submit, and a green pop-up box will appear if the run was submitted successfully. The run should then immediately show up in the My Projects tab. When you select My Projects, it takes you to this page where all the information about your workflow runs is stored. From here, you can search for a run of interest, decide what columns of information you want to include in your project table, refresh your data, and when you select different projects, you can delete projects, share or unshare them with other people who have NMDC EDGE accounts, and publish your project, meaning it would become publicly visible to all NMDC EDGE users. This screen also allows you to check the status of your runs. You can see 4 examples here, the one we just submitted, which is in the queue, some that completed successfully, one that is still running, and another that failed. Here are all of the options for your project status. In order to look at the results for a specific run, you can select this button. For a failed project, you can go in and check the log file to help you track down what went wrong. If a run fails, double-check that your input data is in an acceptable file format. If a run keeps failing for any reason, contact the NMDC team for assistance. For projects that have completed successfully, you can view the results. More information on specific result files and visualizations are found in the workflow-specific tutorials. We encourage users who process their microbiome data using NMDC EDGE to then submit their data and metadata to the NMDC data portal so that it is discoverable for others to cite and reuse. This has been a brief overview on how to log into NMDC EDGE, how to navigate the interface, and how to run NMDC's standardized bioinformatics workflows. The NMDC is community-driven, and our iterative design process for NMDC EDGE is continuously shaped and improved by the feedback we receive from microbiome researchers. If you are interested in providing feedback, please reach out here or visit our user research webpage. And feel free to contact us with any questions, and we would be happy to help. We encourage you to explore all of the ways that the NMDC can support your microbiome research.